So yeah, please welcome everyone. It is for this Interpol webinar, and it is my great pleasure to introduce Dr. Franca Schmid from the University of Bern. Franca is a junior group leader at the University of Bern, and actually she graduated in mechanical engineering, we just heard, in Stuttgart for her master, but then afterwards did a PhD still in mechanical engineering at ETH in Zurich with Patrick Jenny. Then she had several positions, postdoctoral researcher positions, visiting positions at universities in Zurich, Stuttgart, Paris, institutes in Paris, and so on. She's very active also in acquiring grants. Currently, if I understand properly, you are an Ambizione Fellow, sounds very ambitious, from the Swiss National Science Foundation, but also others. Her main work is related to modeling and numerical simulation. So she develops models and numerical simulation tools for biological flow through and transport in particular for situations that go beyond actually what we can see by biomedical imaging. I know there are many applications, so I shouldn't talk too much, but I was enchanted by one you wrote on your webpage, energy supply in brain. In my case, yes, I certainly need to know more about this and how can I improve this. And let's not keep the story too long. She will tell us about stroke across scales. Sounds a bit scary, but I'm sure the science behind is way more nice, and talking about the impact of blood perfusion and oxygen supply in the brain. So please, Franca, the floor is yours.
Yes, thank you very much for the introduction, and also thanking you for having me at this Interpol Academy webinar today. Yes, it was basically during my PhD when I shifted from classical mechanical engineering to apply engineering tools on understanding the brain. And I'm still very fascinated about this interdisciplinary research. And I want to give you some insights in how we can study the brain, and also what we can actually then learn from these simulations in context of biomedical questions. To get started, as I said, the brain is a very fascinating organ, and I want to underline that with a few numbers for you. Let's first look at the neurons. So on the neuronal side, we have 85 billion of neurons in the brain, and these form 1 quadrillion of synapses. And these numbers already show us that the neuronal side of things is very, very complex. It's also very fast because these neurons can fire up to 200 action potentials per second. So that's basically the speed of communication that we have in the brain. I'm not working on the neuronal side of things, but I'm working on flow and transport. But here things are similarly exciting, I would say. So the first number, and which is kind of also a standard introduction if we talk about the brain, but I still think it's very, very fascinating, that you have to imagine that the brain actually is a relatively small organ, but it consumes 20% of the body's oxygen. And what is also very interesting, despite this very high energy demand, the capacity to store energy is actually very, very limited. And this fact really underlines that the supply of blood to the brain really needs to be very robust and reliable in order to avoid that we have tissue damage. To obtain this blood supply, we have about 650 kilometers of blood vessels in the brain. And these vessels also aren't just static, but they're actually also able to locally upregulate flow if we have an increased neuronal activity, for example. And this also happens relatively fast in less than one second. As I said, the vasculature is basically the entity that is responsible to delivering blood to all brain areas, and also the nutrients to the different brain areas. And here you see an image, which is really the seminal work from the 80s of Duvernois, who started to characterize how does this vasculature of the brain actually look like. And here we see it from the outermost layer of the brain, so the surface for the human brain. If we then look a little bit closer, and you see now a slice through the brain, you see these two different colors. So the outermost dark gray part and the lighter part inside. And this is what is called the gray matter and the white matter. And the gray matter is basically the area of the brain in which all the active information processing is taking place. While the white area below, simply put, is responsible for wiring different brain regions with each other. As you can imagine, it's actually not so easy to image this organ. And so in the end, most of our knowledge is actually really limited, let's say, to this outermost layer, the so-called gray matter of the brain. What is interesting about this area, if we now look at the vessels, is that this area has a very hierarchical organization of the vasculature. So at the surface of the brain, that's also again what you see in the image of the right of the Venoy, we have what is called the pile arteries. And the pile arteries, they distribute the blood to all different brain areas. They are then followed by what is called descending arteries, and they really dive down perpendicular into the cortex. Afterwards, we have the very interconnected capillary bed, and blood is then drained in a similar fashion by going up again to the cortical surface via an ascending vein, and then leaving again the brain via the so-called pile veins. And this topology is really specific to this outermost layer of the brain, the so-called cortex. Again, I want to share with you some numbers again. So if we now imagine we have 1 cubic millimeter of brain tissue. In this area, we have about 20,000 of blood vessels, which corresponds to 1 meter of vessels and 100,000 of red blood cells that travel through the system. And I believe that just these numbers already give you an idea that the system we're looking at is actually also very interesting to understand, just given the complexity and the number of vessels we have. If we now proceed basically from the image of Duvernois, who did the first classification of the vessels of the brain, we look at some more recent work. We now see here the vasculature of an entire mouse brain. And as you can see that the data is about 5 years old, and since then there have been a few groups. But now one can really say it has become possible to really acquire, at least for the mouse, the entire vasculature. And of course, this gives huge potential for running blood flow simulations in these networks, and also gives huge potential to generate some new insights of what is going on in a deeper layer and how the blood flow is actually regulated in the brain. However, if we want to understand the vasculature of the brain, and especially also how it adapts to perfusion changes, we cannot only focus at the vessels, but we also need to look at the neural cells. And the neural cells are basically cells that are wrapped around the vessels, and we have different types of them. So for example, around the arterioles, we have so-called smooth muscle cells, while then in the capillary bed, we have so-called pericytes. What all of these cells have in common is that they can respond basically either to certain ions or also to shear stress, and then in function of that change their vessel diameter and thereby regulate blood flow. As I said, this is relevant for blood flow regulation, but it's also relevant if we think about aging and pathologies, because we also know that during aging and pathologies, these neural cells can actually change their characteristics or can also be completely lost. And this again is then a potential cause for impairments in the energy supply during aging. So much about the biological background to make sure we all know a little bit what I'm talking about. So why do we now need in silico blood flow modeling to understand basically what is going on in the brain? Why can we not just, well, do biomedical imaging and look at things? And I think one of the key reasons is really the scale of the common imaging techniques that we have. So a very common imaging technique to understand microvascular flow is two-photon microscopy. And here you can really look in a mouse brain at individual vessels and, for example, record the flow velocity. But the problem really is you can really just look at one capillary at a time. So it's very difficult to get a general picture of what is going on. And another very common imaging technique that is then again used more in the clinics, but also in research, is whole-wave MRI. Now we can look at the entire brain, but we lack resolution because the voxel size in bold fMRI is about 1 cubic millimeter. And if you remember, about 2 slides ago, I told you that in 1 cubic millimeter, we have 20,000 of vessels. So again, it's very difficult to basically connect what happens at a single vessel scale and how does this impact now our perfusion on the larger scale. And this is what I call the imaging gap. And I believe that in silico modeling is actually very, very helpful to close this imaging gap. So in the end, in our in silico modeling, we get a very high temporal and also spatial resolution, meaning that we can really compute the flow velocity in all vessels. And it also allows us to really induce an isolated change and study the impact of this change on the flow field, which again is impossible in doing in an experiment or in a biological setting. Of course, there are also challenges related to in silico modeling because, as you probably all know, we always have to do some simplifications and assumptions. Also, the validation can be very difficult, and thus I believe that in the end, we really need to be aware of these sensitivities and adjust our research questions related to that. Long story short, we really need a strong connection between in vivo and in silico work in order to increase the gain of knowledge on the energy of supply of the brain. And that is really what I'm trying to work on, to establish this connection and then really bring things together to move forward our understanding of the brain. So in today's talk, I want to basically look at 3 sections. So first of all, I would talk a little bit more about the methods we do and how we develop our model to give you kind of a basic understanding of how we can model microvascular flow. And then I want to proceed to some baseline characteristics of microvascular flow in the brain and on some pathological alterations. And so these last 2 sections, they're basically then more related to the research I have been doing, while the first one, let's say, is more, educational perspective on things. Yes, let's start by having a look at the methods and how we can model microvascular flow. So our goal is really that we want to compute the flow field and the red blood cell distribution in an entire microvascular network, as you can see in the image on the left. And in theory, that can be very complicated because, as you can see from this animation here, we have all these red blood cells traveling through the systems and they heavily deform and impact the flow field. And of course, running such a simulation would be very computationally costly. Thus, the question is, to which level of detail do we actually need to go to generate relevant biological insights? And how can we describe the motion of red blood cells in a simplified manner and still allowing computationally reasonable blood flow simulations? And the first thing that you commonly do in fluid dynamics in general is to have a look at the dimensionless numbers. And here I listed the ones that are in the end relevant to us. So of course we have the Reynolds number, which gives us the ratio between inertial and viscous forces.